Welcome back to the Biostasis Technologies podcast. Our guest this month is Ted Brown, who amazingly has an even longer history in cryonics than I do. Been around for a very long time and is therefore fairly hard to introduce because there's so many things we could talk about. So I'm going to let Ted tell us about his involvement himself, starting off in the very early days in the New York area. So Ted, tell us about how you first got involved in cryonics. How did you hear about it? Who were the first people you met and the interesting characters, and how did that get started?
Yeah, I've had a long-standing interest in cryonics. I think I got interested in it when I was a high school student, and I read about Edinger coming out with a book, and it was in the 1964 Playboy magazine, and Frederick Poole had an article on immortality prospects. So it got really interesting, and so I read up on cryobiology, and I read that you could freeze animals like rats and hamsters to the point where 80% of their water was ice, and then you could bring them back to life. And I thought that was pretty amazing. So I got some pet rats and put one in the freezer for an hour and a half or so and brought it out, and it was dead. And I put it under a heating lamp for 10 minutes, and it started coming back, and it came back to life. I was pretty amazed. So I thought, well, that's really exciting. And I thought more about it and thought, well, I should become a doctor and help freeze people. And that's why I went off to, I applied and got into Johns Hopkins as an undergraduate. And that was back in 1964, 65.
Wow, just when I was being born. I think you mentioned to me before your mother died when you were quite young. Is that right?
Yeah.
Did that have any effect on your interest, do you think?
I think I was always thinking about death and dying since she died when I was 6 years old of pancreatic cancer. She was 42. So, it was something on my mind. Where had she gone? How would I ever see her again? And, it led me to think a lot about the brain and the mind and what we are as as conscious people. And in high school, I kind of decided it was pretty unlikely I would see her, and thought more that got into science and biology and finding that much more interesting and thinking about life and all it means. And so I thought that's sort of how I got interested in the idea of freezing in the long term to come back in the future and not have to die. And so that kind of is what I got to Hopkins as an undergraduate, with the idea that I would eventually become a doctor and be a freezing and help people to freeze. And I got in touch with Ev Cooper, and I don't quite remember how I met up with him, but it was during my first year as a freshman, and I became involved with him. And Ev Cooper was probably the first cryonicist. That's at least what Mike Perry said in his write-up about him. Ev had been born in Butte, Montana. I was in Missoula, Montana, but Butte, and he had been born to a mining magnate. And shortly after he was born, when he was 3, they moved to Seattle, and the father, I think, left mining but was fairly rich. And Ev grew up there and went to the University of Washington, but he was named Stanley McBain at the time. And then he got married and moved to Oregon for a while and went into the Navy during the war, and eventually ended up in Florida and apparently was in the Navy and enjoyed sailing. Somehow he and his wife divorced, and then he changed his name to Ev Cooper and moved to Washington, D.C. And he was a very philosophical guy and wrote an article. He wrote a book about immortality, physical, possibility, scientific possibility now, and published it in 1962. And then he started a group of people interested in immortality and the physical reality of that being possible.
That year, 1962, I think, was the same year that Edinger published his book unofficially, right? Like self-published.
A version of it. Then this group that Ev started, we were talking, they'd been meeting for a year or two, and then they started talking with Edinger, and so he invited Edinger to come and meet up with him, and they talked it over, and so it was Ev's book came out first. He published it himself, and then 6 months later they got a copy of the preprint, but it wasn't until later that his book was actually made available publicly till '64, I think.
Hmm.
But actually, Edinger became the representative for Detroit of the Life Extension Society that Ev had started, and I was the Baltimore coordinator. And Ev came up, and we had a meeting at Hopkins one time, and invited faculty members to come, and it was written up in his newsletter. And he would come out with a monthly newsletter, and he did this for 5 years, and it was called Freeze, Wait, Reanimate.
I've always liked that. I know we don't freeze really, we vitrify, but still, I always like that tagline.
Right, right.
Yeah.
And I remember going to visit him several times, and we would go sailing on his sailboat, and then he was building a freezing facility in a remote part of Maryland, and it was out of bricks, out of big cement bricks and mortar. And I went there one day to help him a bit with it, though, and could see in the picture of the newsletter what it was looking like. And he kept asking for people to help out with it and join up, and I could see that it was they had apparently bought the land, and then they had to get a mortgage on it, and it was $50,000 to build, which was a lot of money in those days.
Yeah.
And then I was very involved with my studies at Hopkins, and I was graduating in record time in 3 years' time. So the Vietnam War was approaching, but I got into graduate school and I wanted to study biophysics, thinking that was close to freezing and understanding about it. And at this time, I kind of lost touch with Ev, but I know that then they had the first freezings in California. And Saul Kent had met up with Ev, and for a while was a coordinator in New York, but then he started his own group, and then he and his colleagues went to California and met up with Nelson and others, and they froze Marie, and then they froze Bedford, and somehow Edinger and Cooper got into a disagreement, and they had a falling out, and it had something to do with Cooper thinking that it was all fraudulent. Cooper's idea had been you really need to get big companies involved with cryonics to make it a going, and he was very disappointed that it wasn't really taking off, and then he had this disagreement with Edinger about Nelson, and he thought it was all fraudulent, so they kind of had a falling out.
You thought the Nelson was fraudulent, not cryonics.
Yeah. But it's just Edinger, and he kind of fell out of touch, and I was kind of unaware of this. I was doing my studies, and then I got into I spent 2 years in graduate school in biophysics, working on my PhD. I had been taking courses, and then my draft number was coming up to go back to Montana to the Vietnam War, and I thought it was a good time to be applying to medical school. So I got into Harvard Medical School, and went up to Boston and started medical school. And my first year there, I went over to the freezing laboratory, the cryonic laboratory at Mass General Hospital, where they freeze blood and tissues and embryos and things. And I did some experiments with Charlie Huggins, who ran the lab, and kept reading up on what it would take to freeze organs, and it seemed to me that was kind of a very technical problem that would be solved before too long. It wasn't all that interesting.
Yeah.
But I thought the problem would be once you come back from being frozen, you'd still have to deal with aging.
Mm-hmm.
And I got to thinking aging, was a more interesting problem, more challenging. What causes it, and could we slow it down? And so I went to talk to different people, professors at Harvard, on what they were interested in aging, and I got in touch with John Little, who had a lab at the Public School of Health, and they were doing research on aging in progeria cells. And I'd seen a case of progeria on TV and thought that was really an interesting model of aging. So I started.
Could you just explain progeria for those who may not be familiar?
Progeria is a rare disease, a genetic disease that looks like premature aging in children. And the children that have this are born looking pretty normal, but they develop an aged appearance by age 1 to 2, and look like little old men by the time they're 8 to 10. And the average lifespan is 12 to 14 years, and they die of heart attacks, and they're quite and dwarf, but they look like they're barely look old, senile men as they grow up. And it's quite a striking disease. The frequency of it's about 1 every 4 million births, so it's quite rare. And any one time in the United States, there are only about a dozen or so cases alive that have been born here. We know, and so it's a rare disease. Anyway, we'd gotten cells from patients, skin biopsies, and we're growing them, and we're studying DNA repair. And so I thought it was something I could pursue. So when I got graduated from medical school, came to New York to do training in medicine. And after 3 years of internal medicine residency, I was trying to decide to go into geriatrics or gerontology or what. But I thought I'd just stay with genetics and study progeria. And I'd gotten a grant. I got a grant from NIH to study progeria, but it was very hard to study. A very rare disease, and it's not inherited. It's new dominant mutation, sort of like achondroplastic dwarfism. Short-limb dwarfism is usually not inherited, though it can be, but it can be passed on once the mutation is present. And it's the same case with progeria. Neither parent is a carrier, but they have a child, and that child has this rare mutation. And these children they're infertile. They're dwarfed, and they don't pass it on. So it's never inherited. So it's a new mutation. And in those days, in the 1970s, it was very hard to find a mutation like this, a new dominant mutation. Anyway, I was looking for a biomarker, and I'd gotten a grant to study biomarkers. And we looked at DNA repair. It was not too reliable. We found, missing HLA antigens or rapid protein deterioration, and none of those seemed to pass. I couldn't really validate those with my work. So I kept interested in progeria, but I turned to working on Down syndrome as a model of aging, because they have early Alzheimer's disease and they have a premature aged looking phenotype. And
let's stick with progeria for just a second. Do you think the progeria is a good model to study for aging, or do you think it's kind of a...
Well, that was the reason I was studying it, thinking that it might connect, and we still kind of think there might be an overlap, but we've discovered the cause of it as being a mutation, a new dominant mutation of lamin AC is the gene, and that's a gene that makes a protein that is abnormally farnesylated. Has a lipid group that doesn't get it removed because of the mutation, and that causes nuclear distortion and rapid aging of the heart and atherosclerosis. But it doesn't affect the brain for some reason, because they have a protective microRNA, it appears. So it's a model for aging. But do we all have the abnormal protein? Probably not. But we often develop a little bit of it. So there's always been a question of how much of that is really aging or not. But it's always just been a striking model of aging. So that it was kind of something I would keep in touch with, and I would try to accumulate skin biopsies of the patients so that I could study them in the lab. And after a few years, I had been in my fellowship in genetics and became an assistant professor at Cornell Medical School. And the department was disbanding. The head of the medicine was leaving and retiring. And so I decided I would go to Rockefeller, where I got a position for a short time to do, genetic work. We were going to sequence the 21st chromosome. And then I got an offer to go to Staten Island and be head of a laboratory of genetics and be a department chairman of 5 laboratories. And so I took my work on progeria and Down syndrome with me to this institute, the New York State Institute for Basic Research, and became the head of the department there for genetics. And while I was there, I met a family that had a progeria child that was about 2 years of age. And they came to see me to get it verified, and I confirmed the diagnosis. And the family were both doctors, and they decided they'd set up a foundation to study progeria. And the mother, who had been in a PhD program, gave up her residency to work full time on it. And the family started the Progeria Research Foundation, which I helped out with and became very involved with. And then we had a meeting at NIH on progeria. The first meeting kind of organized of anybody that had published on it or had any interest in it. And it was Francis Collins came, and he'd had a child that had a variant of progeria when he was in his medical training. And so he thought it was quite interesting, and we decided we would set up a consortium to try to find the gene with his help. And for a year or so, we had this consortium underway. And one of the cases I'd seen had a rare chromosomal abnormality. So we thought we could clone the ends of this translocation, of this inverted insertion. And it worked out that while we were doing that, and it was located on chromosome 1, that there was a case of a rare disease that's sometimes confused with progeria called mandibuloacral dysplasia. And it's a hereditary recessive disorder, but looks something like progeria. And it was on chromosome 1 and it affected this gene, lamin A. So he was able very quickly to get it sequenced and we found the mutation in these cell lines that I'd been accumulating and published and found the cause of it. And that led to discovery that there could be a treatment for it. A drug that had been used for RAS mutations was available, and that's been used in trials of the children. And there's still a lot of work on it. I still am involved with the Progeria Foundation and research is underway. There are attempts underway now to find a genetic therapy to correct the gene mutation, but it hasn't yet quite got to the clinic yet. But we're very close to it, and Francis is still involved with it, even though he's retired from being the head of NIH. He still runs a lab and is working on that with other groups, and so it's close. We had a very nice meeting last fall on progeria, and it's every 2 years we've had a meeting. So it's kind of been a model of aging, something I've been always interested, but I don't think it tells us much about true aging.
Right. You told me that, and I was surprised that you did some AI research in the mid-1960s, which is very early on. How did that fit in with the progeria and Down syndrome and other work you were doing?
Well, when I was in the graduate school in biophysics, you had to do you had to be a training, had to do a lab teacher. You had to be an assistant in a laboratory, and I was interested in chess. I'd played chess when I was in high school. It actually won the state championship, Montana State Championship, when I was a senior. So I thought it'd be fun to program a computer to play chess. And in the biophysics department, they had a small PDP-11 computer, and the professor that was using it to study the brain, and I'd been interested in brain function, so I got in touch with him and started working on that as my thesis project. But then I found out that they had a course in artificial intelligence that I could be the graduate instructor. So I went over and arranged to do my teaching on that. We programmed computers in those days with Fortran and cards to play BASIC, using BASIC as a language to play.
This would be back in the days of Minsky, McCarthy,
that's right.
Neumann, those kind of people.
Yeah, that was fun. And actually, when I got to Harvard, to medical school, I was able to I had some extra time, and I went to MIT and took a course on artificial intelligence. I think Minsky was one of the professors there. And it was, I enjoyed it. It was fun to think about that. And I still think about it. I mean, it's still on my mind that we're really facing, artificial general intelligence is not too far in the future. And it's really going to change everything.
Are you sure about that? I'm very skeptical about the current approaches to AI. I think the LLMs are not going to become super intelligent, not going to be AGIs. I think they're going to hit a wall. But that's not a very popular view right now.
Jeffrey Hinton, who's got a Nobel Prize for AI and kind of the father of AI, came out with a statement that I read a few weeks ago that, the power of the progress in AI is doubling every 7 months. We're getting more powerful. So if you figure exponentially every 7 months, in 2 years it'll be 10-fold stronger. In 4 years it'll be 100-fold stronger. And think of having a computer that's 100 times stronger than the ones we have right now, or your phone 100 times stronger. what it can do. And that's really what they're envisioning, that you need larger and larger, stronger and stronger computers to be able to generalize these language models and encompass all of the information. So I think, and also the book, like Ray Kurzweil's book, envisions within a short time we're going to achieve a much, a general intelligence is going to progress to that point where we have, they're going to be smarter than we are.
But the trouble is these systems don't work like human brains, and they're not going to scale a lot longer, right? Running out of training data, they're using massive amounts of electricity. It's not at all clear to me that the progress that we've had in the past, it's easy to make these extrapolations, and people love to do these with singularity discussions. But does that mean that's actually going to happen? I think we'll know within a couple of years, because there's so many people who are predicting an AGI in the next year or two.
I mean, it is
5 years from now, it hasn't happened.
It's really incredible how fast it's increasing. I mean, NVIDIA, the way it's gone up.
Yeah.
All these, billions of dollars being built for these data centers, and yet.
Well, we'll see. I'm not so sanguine that they're actually going to become super intelligent and solve problems for us. But
well, I'm not sure it'll solve the problems because there's a worry. I was growing up worried about having a nuclear war and wiping out the future, and this is an equal kind of worry. Is what is the prospects of these robots taking over once they get to be that much smarter than we are.
I think it'll happen eventually. I don't think the current approach is going to make it, and I think it's all the money's going into the current generative AI approach, and everything else is being starved. So all these alternative approaches, which were discussed actually back in the day, the 60s, 70s, 80s, sort of the good old-fashioned AI, as they used to call it. Nobody's doing that kind of stuff anymore, hardly anybody. So, but we'll see. I guess events will soon show it one way or another.
Well, just to go back to kind of tie up with Ev Cooper, so I kind of lost touch with him, and he kind of lost interest in cryonics after 5 years of publishing his newsletter, and he divorced his wife and he moved to Connecticut to go sailing, and for 15 years he was just a sailor, and then he was taking his boat to go to South Carolina, and he got lost in a storm, we think, and was lost at sea. So he never got frozen, and he kind of gave up on the idea after that fallout with Edinger early on, I think. But he was the first. He was the first cryonicist.
That's too bad. The first two people didn't get on better. That was very unfortunate.
Yeah.
Okay. So let's see. After that, so we skip quite a few years later, and you did some work in Australia, right, with Southern Cryonics, the first real cryonics organization in Australia.
Yeah, so I, after working as a geneticist and running this institute for some 30 years or so, and focused on, causes of intellectual disability and what we could do to treat that, Fragile X and Down syndrome and autism were the kind of topics I worked on mostly. With it, still had an interest in progeria, but mainly focused on intellectual disability. I retired and moved to Australia to get remarried to a pediatrician that I'd met, and
this is about 10 years ago.
Yeah. Just about 2016. I retired. 2017, I got remarried in January, and then so I read an article in the New York Times about the cryonics movement again, and what's going on, and they mentioned there a Southern Cryonics group. And Peter Tokalides had set it up. So I got in touch with him, and they were having a membership, it's closing, that the first 34 people would be primary members. And I joined up with them, and they had just about finished their facility. He worked with Phil Rhodes, who's very involved with the cryonics there, and they had Aaron Drake come down to help train, but because of COVID, it was a problem getting in and out of Australia in that period of time.
Aaron Drake at that time was medical response director at Alcor, just for the background.
Yeah, but he was very helpful to Peter, and Peter had come up to visit Alcor and Cryonics Institute to kind of figure out what was good programming.
Peter's timing was excellent. I think I mentioned this before when we talked, but Peter came to visit at a time when we had a cryonics case going on, and also very, very rarely had a delivery of a cryogenic duo, which doesn't happen very often at all, every couple of years. So he got to see everything. He was very lucky.
Oh, great. Yeah. Yeah, well, he's a very bright guy and engineering type, and worked with Exxon and knew what he's doing, and he was very dedicated to getting this facility going, and with a group of 30 or 40 people there in Australia that had become cryonicists and involved. So I was very happy to join up with them. I didn't have too much to do with the facility, but I was just supportive of their efforts, and then it wasn't working out with my marriage, so I came back here and found out about what was going on in New York, and I got in touch with Ashwin, and I think that's where I met you first. I came to one of the meetings in Williamsburg a couple of years ago, and that's where I heard the talk from about Tomorrow Biostasis, and Eric, or the head of that, gave a very nice Zoom.
Emil Kinziura, yes.
Yeah, Emil, kind of hard to remember his name.
Kinziura.
Yeah, Kinziura. Yeah, and I was impressed that he'd been a medical doctor and interested in aging, but kind of gave it up to work on cryonics is the main plan. And but I was impressed by his enthusiasm and what they were doing, and I kind of got thinking. I'd been thinking I'd come back and join up with Alcor, but I'd kind of found that there'd been a lot of political, dissension with people at times, and I wasn't quite sure what was going on there. It didn't sound all that attractive to me at the time, so I felt I really should think about, Tomorrow Biostasis is where in my plan. So I got in touch. Ashwin had recommended that I talk to Rudy Hoffman, and I got in touch with him, and they would help me set up my trust fund and account to get it established so that I'm all paid for to go to Switzerland when I'm frozen, and to be stored there in their facility. And
so what did you like about Tomorrow Bio? Why did you choose them, even though they're not
well,
based in the US?
It seemed like that they were really growing, and they had a lot of young enthusiasm, and they knew, and it seemed like they were on top of all the procedures, and were doing a really good program. I mean, it just seemed to me is an alternative to Alcor, and it was either one would be a storage place, and a question of how you'd get there. And so I learned about SST, and I went to one of their training sessions, and that's where I guess I learned. I became one of the EM people watching over the procedure and helping out and learning what I could do. I mean, I've helped out with the one case a bit, but I'm not really haven't been involved in it from the first stages of a live case. But I'd be glad to. I'm still a registered doctor here in New York, a physician, and but I'm still very interested in aging and longevity, and that's one of the main things I try to keep up with. And it was it's quite amazing that Tomorrow Bio, they had a video tape. They had an announcement on February 18th that they're going to now provide information on longevity to all their members. And it seems like Emil's coming back and kind of focusing more on, what they have over 1,000 members signed up. The average age is 37. So for the 40 years they're waiting until they're old enough to be put in storage, what are they going to be doing? So they're thinking, well, they should help them, with longevity and living their best lives and staying alive as long as they can.
Yeah.
And things may change. I'm optimistic that we're going to find out what causes aging before too long, within 5 or 10 years, I'm estimating that. And we really need to understand what we can do to stop it. I mean, if you look at, like reptile, like tortoises don't age. Their risk of dying is about 1% a year from juvenile to their 150th year.
Unlike sharks, right? Sharks don't really.
And sharks are the same. Yeah. There's something, and but we have genes. We live to be 100. Mice live to be 3. There's a genetic difference. What causes that genetic difference?
Naked mole rats live a lot longer than
they live 20 or 30 years, and they have big their hyaluronic acid is very high level in their body, And bats have all these DNA repair enzymes that we don't have that keep all these viruses under control and live a long time. But what is the difference between a tortoise not aging and a human that ages, or most animals? And there must be a difference that we can understand and manipulate and correct.
So do you think there are any current effective life extension treatments, things that will actually either greatly reduce the risk of premature death or actually extend maximum lifespan?
Well, I know, I mean, the one drug that has been found to extend lifespan in any animal where it's been tested is rapamycin. It's the drug that's been around for 25 years. It's kind of an immunosuppressant drug, but it's been found, if it's given properly, it'll extend lifespan in any animal that's been tested in. And they're testing it in dogs and marmosets. There haven't been any long-term studies in humans, but it's underway. But I'm one of about 3,000, maybe not 30,000 people we estimate that are taking intermittent rapamycin. I take it once a week. And that suppresses the aging, the mTOR1 system in the body, and that seems to prolong lifespan in animals. So it probably does in humans as well. We think that mTOR1 is kind of the main nutrient sensing system in the body. If you're taking in food, it gets active and it causes growth and maybe cancer. But if you go into a starvation, it inhibits it, and you go into a survival mode. And that's kind of what you're doing when you're inhibiting it, is surviving under that. So that's, but like,
well, that's interesting. It's interesting because if you think about calorie restriction, that seems to work across species, but it doesn't seem to work very well in humans. So
yeah, but there may be a better drug for that. And I mean, like Matt Kaberlein has a company. He's a gerontologist at the University of Washington, quite well known, and worked on rapamycin a lot. And he's set up a company, Aura, and they're looking at thousands of compounds to see what's a better drug than rapamycin. And there's a webinar that he has out. It's a proprietary announcement. They found one that's 2 or 3 times better than rapamycin at this point. So I think progress will be made there. There is progress. I mean, I've always thought that the way that cryonics would be accepted better is if we could freeze organs, and that was my idea when I was back in medical school. That was what we needed to do. So there's a company that Laura Deming has started, where they're until.com it is, and they've gotten $50 to $100 million to perfect organ preservation. And there's a company, Loyal, that's doing a trial of a drug for dog aging and improving that, and then methods for organ. If you can show that it'll work for animals, and if we can then apply it, like for freezing kidneys, then I think the medical profession will accept that we could be frozen and come back in the future.
It should make a big difference, you would think, and it's been encouraging that we've seen some recent breakthroughs. It's been very slow, obviously, over the decades, but then the University of Minnesota, we've seen some repeated success with rabbit kidneys.
Right, rabbit.
Actually, no, it was rat kidneys, sorry, at the University of Minnesota, but rabbit kidneys as well.
Yeah, I mean, Greg Fay's work and Mishkoff in Michigan and Risco in Barcelona are, here and there are people working on organ preservation, and that, but it's not that far off. But there's still a challenge.
Yeah. So, is it just rapamycin? Do you think there's anything else that you think is interesting?
Yeah, there are other things that are. I mean, there are 150 biotech companies that have been started, and it's just mushrooming, blooming, and each one has their own idea of what could be done to help with aging or improve longevity. I mean, Jeff Bezos put in $3 billion with his colleague to start Calico or something, a company in California,
Altos.
Where the idea is regeneration using the Yamanaka factors. And it's, Sinclair's group is trying it out. They tried it out with an eye problem, and they're now trying. It's been FDA approved for an eye treatment, and if it works there, it might rejuvenate. It might be used for whole body rejuvenation before too long. I mean, there are all these different avenues that the field is exploding with interest in longevity medicine.
But these are all things like in the research lab. Rapamycin is something you can actually take. Do you think there's anything else that, in your view, is worth taking right now, or depending on
Well,
your
well,
age?
yeah, yeah. I mean, actually, there are several doctors that have blogs and websites, and both of them have a high genetic risk for Alzheimer's disease. The main gene for Alzheimer's risk is ApoE, apolipoprotein type E, and if you have type 4, you're more at risk than type 3, and then 3 is more at risk than type 2. Most people have 3, and about 25% of the people carry 4. But some people carry 2 fours, like the actor Chris Helmworth was found to be a 4-4 carrier.
Bad
And that
news.
increases his risk of about tenfold of getting Alzheimer's disease,
Mm,
often in their 60s. Uh, and so there's a lot of interest, and they have blogs, and they're trying out different drugs or talking about things. So I've been following along with these. I've started low dose lithium, which I think is very helpful for brain. It's and avoiding Alzheimer's and keeping your
mhm.
brain young. And low dose doxycycline may improve the blood brain barrier, and other things of that sort that are medicines, but also just supplements. And omega 3s are very important for the brain.
I was
In,
just reading something this morning actually about visceral fat and the effects on brain aging. So apparently, it's tricky looking at correlations between BMI and various problems because there's a lot of confounding factors, like people with Alzheimer's tend to lose weight and so on. But
right.
it seems like there's pretty strong evidence that if you've got a lot of visceral fat, that's really bad for your brain. So
Yeah, yeah.
doing the exercise, getting that weight off, and doing the exercise to get rid of visceral fat is probably an
And
important
there
thing.
look like there's some drugs you could take to convert that visceral fat to brown fat, and the brown fat releases more pro longevity cytokines and factors that would be helpful. So that's kind of like one of the strategies that are
What
kind of
about metformin? Do you take metformin?
I've been taking metformin for about 10 years, even though I'm not a diabetic. But it has somewhat of a similar action to rapamycin.
Mm-hmm.
It's still not as good as rapamycin, but it probably has in animals it prolongs the lifespan somewhat.
Yeah, I've been taking metformin for a while. I stopped briefly because I heard some suggestions that it might actually be bad for your brain, but that didn't seem to follow through.
No, I mean
And
there's
other
some
people saying it might block muscle
it yeah it,
development.
it,
But I think
it
that's largely theoretical too. That doesn't seem to really
yeah
be a
yeah I
feature.
think if you exercise you keep your muscles in pretty good shape, and that's very important to exercise because that also releases good growth factors and
Mm-hmm.
keeps you young, your body young as much as possible. And that and sleep and diet and maintaining good friendships and sleep and keeping your brain stimulated with
All right. But still, the
with
core things,
junk.
right? All these interesting things in labs, but it still matters is sleep, exercise,
That's right.
diet, and,
That's
and
right.
community. Yeah.
Right, right.
Yeah. Is there anything you do apart from that which is maybe a little unusual that people would be interested in?
Well, I play poker. And I have a game tonight, actually, a tournament. I happened to win the game tournament last week, so it's exciting when I can play poker. I play chess and
Mm.
chess problems every day. Unusual. I play the piano. I studied piano for 10 years and violin for 4. Um, don't play very much anymore, but I have a grand piano here. Um, I'm
So
not sure
cognitive diversity, keep your brain doing different things.
what.
Cognitive diversity, keep your
Yeah,
brain doing different
yeah, yeah,
things.
yeah. But I spend a lot of my time just trying to keep up with the literature on aging and longevity because there's so much coming out.
Do you have any recommend? Yeah, that is a problem. There's so much stuff out there. Do you have any recommended sources? There's some interesting, online resources these days. Is there anything you particularly would recommend people keep an eye on for good, solid news, critical thinking?
Um, well, I mean, this Nick Notwich is a doctor that has the double ApoE4 allele, and he has this twice a week. I get a newsletter from him, and it's always he's a young MD PhD. He's got his degree at Harvard Medical about a year ago, but he got a PhD at Oxford in metabolism, and he's really sharp and takes. And so I find him to be very helpful. I follow there's a rapamycin administrator a newsletter blog that comes out twice a week or more often, and it has a wealth of information about aging in general, but about rapamycin and
Hmm.
people that are on it, intermittently. Um, and I think there's Brian Kennedy's group in Singapore has every twice a week, a talk, and he's a very good investigator in aging. Um,
Well, he used to be at the Buck Institute, right?
he used to be the head of the Buck Institute.
Yeah.
Yeah, after he stepped down from that, but the Buck Institute's doing good work. They have Burden is there, and and there are an increasing number of longevity meetings. It's just hard to think about all.
This morning I was looking at an article. I was reading something else and it popped up. It said 25 best longevity conferences this year. There's just too many. I mean, it seems like a lot of them seem very repetitive too. You see a lot of the same speakers and the same stuff.
Yeah.
Some of them, of course, more focused on investing side of things.
Yeah,
But,
there is that. I mean,
it's hard to find reliable information. That's the thing. That's why I ask you if you have any favorite sources. Like longevity technology seems to be a fairly good sort of journalistic reporting on advances.
yeah,
On the other hand, you have people like Brian Johnson, who I think is getting a lot of attention these days, but it is not really a very scientific approach. It was all very personalized experimentation at great expense. But what is that actually telling us for the general population?
Right. Yeah. I think there's Peter Diamandis he started these fountain clinics, Fountain Life, Foundation Life, and they have 4 or 5 clinics, and they're doing a great job, but they're very expensive. You have to pay $50,000 to $100,000 a year for their oversight. But
same with Peter Attia, his clinics.
Yeah, it's similar to Peter Attia's clinics. I followed Peter Attia for a while, but I got kind of bored with his long talk, his long interviews of people that didn't give you much new information. But when his book came out, it was quite helpful to read. And he takes rapamycin. Matt Kaberlein takes rapamycin, and it's sort of like it's known that. And Brian Johnson was taking it for a while, and then he quit. But I didn't quite understand his rationale. But it's something that I'm convinced is probably a good, will slow down my aging.
Yeah.
I'm turned 80 this week, see, so I did a face analysis and it said I looked to be 64.
Yeah, doing a good job. Yeah, 80 is the new 50, I'd say.
That's right.
Eventually, 80 will be the new 20, but not just yet.
Right.
So, in terms of getting cryopreserved, well, let's say your optimism doesn't work out and it takes a little longer to fix the aging problem. To me, one of the biggest concerns is getting cryopreserved under good conditions, with the brain intact and without a long delay. Do you have any measures you take to try to minimize those problems, like where you live, or notifications, or the people around you, if they're supportive, that kind of thing?
Well, all my kids, my family know that I have set it up to be cryopreserved. I carry, a band from Tomorrow Bio and a necklace that says who to contact.
Mm-hmm.
If I were sick with cancer or something, I would probably move to be closer to the facility where I could be taken care of quickly. But it's hard. Your life you go on. You have to drive your car places, and that's the biggest risk is having an accident.
Yeah.
But just staying in touch, having a good doctor, and being able to get frequent laboratory tests to show that your lipids are staying in good shape, and I've had a few medical problems. I developed atrial fibrillation and 3 years ago, and then I've had 2 surgeries, but it's gone away for now. But it's kind of a problem with aging that these things crop up. I don't have any serious thing, and I don't have cancer. That's always a risk, but I think it would actually be beneficial to have a disease that you knew you could die from relatively soon, and so you could plan your own death, and that would be the optimal way to be cryopreserved, quickly.
Right.
And,
Yeah, that's kind of, as we were sort of discussing this morning in an
yeah,
online call, it's kind of funny that for cryonicists, the best way to go is a long, lingering death.
that's right.
Because then you can predict the end. It's not like a heart attack or a stroke that comes out of the blue and there's no warning.
Right,
So, yeah,
right,
it's kind of funny.
right.
Grimly funny.
It is.
So,
Well,
what do you think your priorities will be for the next few years in terms of life extension and biostasis? Are you
well,
looking to help with cases or give medical advice or help
I'd
with?
like to help out with the group here in New York with Ashwin's facility here, and I've helped out. I've gone to several of the trainings, and I know um, Aaron Mindel is kind of the operations guy, and Lauren Fusco, who's second helping out with Ashwin. But there's not too much I can do. And it hasn't been too active. We've had several cases, but it's not like there's a lot of need, for a medical doctor. Though I'd be glad to help any way I could. Um, I mean, I'll just stay in touch with Cryonics and probably try to go to this meeting that was mentioned in California in May. It sounds like that would be very interesting. Uh, and it's just I'm kind of coming back into the field, having been away from it for some period of time, but always knew that this is what I wanted to do. Uh, so I haven't really made
Maybe get involved with Tomorrow Biostasis with this new service. I'm curious who they're relying on for their advice, because obviously it's very important to have the right kind of people.
Yeah, yeah. Well, they're still very centered in Europe, but they're trying to expand here to the U.S. And they have they're made, but they don't. I'd be glad to help out, but haven't been asked any. But
Well, they may not know your background there, so you may ask them, because I did ask during the discussion a couple of days ago when that was announced. I asked how are you choosing the people you rely on for the advice, and I think the answer wasn't really satisfactory at that point. I have to push a little more on that, but maybe one of those people they should be consulting.
Right. Well, I'd be glad to help if I can. I mean, I've always admired your work, and I've known you've been very involved with Alcor at the time, and to be a philosopher is just amazing to me because it's quite different than what my background is. And a transhumanist, I still don't quite understand it, but it's something I admire a lot. I think that's what you're doing is great. And you say you're coming out with a new book before.
Yeah. Yeah, as soon as I can get it published, it's finished. I'm just working around agents right now, trying to get them to pick it up.
Good luck with that.
Yeah, I might need it. Well, if I don't get it picked up by a publisher, we're going to self-publish, but I have to try the traditional route first. It's like, Charles Platt was talking about this morning in the call that you were on. And, he wrote most of a book on cryonics, actually at a time when there was quite a bit of public interest in it because of various stories. And he couldn't get it published, so it's just a tough sell, but we'll see.
Yeah. Well, I mean, I think that's been you've been aware of that with Alcor, but that's been the issue is that there hasn't been a general acceptance of it by the population or by the scientific community, and that I think that won't happen until we can prove that you could freeze a kidney and use that for transplant and show that it works. And that shouldn't be that difficult. I mean, it shouldn't. I mean, it's not conceptually. You're just
conceptually, but in practice, it turns out it's quite difficult. Yeah.
Yeah, I mean, it's the warming problem is a problem. We have to use magnetic particles to use the heat to get it all heated rapidly and go. I know it's a problem, but it's just there hasn't been much money put into that area of research, and now that looks like there will be with Laura Deming's companies and
is that the same? Her first company was called Cradle. Did she change the name of that to?
I think so. Yeah, it's now. I think it's now until.
Yeah. I'd like to get her on the podcast actually. And see
yeah,
what she's doing.
I mean, she's apparently a brilliant lady. I haven't met her myself, but she's really an outstanding, young lady star, and started several companies already.
Yeah, someone who can bring money in. That's always a good thing.
And then Alcor, I noticed in one of their news releases, got a big donation from Marlene Roth.
Martine Rothblatt.
Yeah, and apparently,
Yeah.
that helped them out a lot.
Yeah, they must have had a number of donations because they seem to have been able to hire quite a lot of staff recently. So
Yeah,
hopefully
yeah,
that's sustainable. I don't know how the finances are working on that. So I was
yeah.
actually trying to look at Alcor's financial statements, but they're about 3 or 4 years outdated, so I can't
Oh,
actually
really?
tell how they're funding this right now.
Yeah, yeah,
Yeah, but more research certainly good. More research personnel is, it's impressive that Tomorrow Biostasis has like at least a dozen staff right from the beginning. So Emil did a great job in raising funds for that, and I think this
yeah,
new program, which I'd like to know more about, but it sounds very interesting to me that it increases the value of a membership. And really,
yeah,
this is something Alcor should be doing. And I wish I'd done it when I was still there. But Alcor Life Extension Foundation, they should be doing life extension advice as well, and not
yeah,
just at
yeah.
the very last moment.
All right. Well, I'll try to help them out if I can, if they want me to.
Yeah. Well, anything else in particular you want to talk about here?
Um, Not too much. No, I think we've covered much of what we had.
Yeah.
I just think that Alzheimer's is a major risk for many people, and as we get older, it's and a lot of money is still being put into it. I mean, the National Institute of Aging's budget, maybe $10 billion a year, has gone to aging, to Alzheimer's research, or maybe I don't know the figure, but there's a lot of progress being made, and I think that could lead to improvements just in brain health, because that I mean, things that I've been reading about will indicate that. So I just think that's something that we need to keep an eye on, and I think it's very close to coming up with some more definitive treatments. I mean, there are antibodies that are available to slow down the rate of development of Alzheimer's once you have it. It doesn't stop it. It's not a cure. There should be some way to cure it, and that's
Yeah.
what I think we're on the threshold of that too with some of
Well, I think
these.
there's a lot you can do to reduce your risk if you start earlier on, is that people leave it till they've already developed it. And of course, now
Yeah,
there are tests that can suggest 20 to 30 years ahead whether you're going to get it. So
right, right,
you can reduce inflammation, do more exercise, take care of your brain. There's a lot of things you can do, but not if
right,
you leave it too late.
right. I mean, there's a professor at MIT that her lab had been working on ultrasound or gamma wave sound, and they find that gets rid of the amyloid in the brain in mice. And they have a cognitive company that's been set up, cognitive therapy, I think it's called, and they have a trial that's underway. It's in a phase 2 or phase 3 trial now for early Alzheimer's, and I'm optimistic that might turn out to be beneficial.
Isn't there a lot of controversy though right now that suggestion that the dominant amyloid hypothesis might not be correct, and there's some publication issues there, and I don't know how that's turned out recently, but that maybe we've
Yeah,
been pursuing the wrong path or an incomplete
no,
path for a long time.
my understanding is that the, the pathology is that amyloid develops, and it's not necessarily toxic, but what it does is it causes tau to get phosphorylated abnormally. So you get phospho-tau, and tau is what causes nerve death, neurofibrillary tangles. So it's kind of dependent upon the amyloid that causes tau. So they don't have good tau antibodies, though they're working on them. But the mechanism of going from amyloid to tau, because you can have a lot of amyloid and still not have Alzheimer's, but it's the tau that is really damaging, and so that's kind of where the thinking is now. And there are the new blood tests, phospho-tau 217, that's been approved by Lilly and by a Japanese company that has been testing it for spinal fluid, but now you can do a blood test, and these are now LabCorp and Quest are going to be offering these. It's early diagnosis, you're screening, but the mechanism is that the amyloid isn't primarily the cause. It's something related to it causing tau to get phosphorylated, and in my understanding. So if you could prevent that
Yeah.
connection, then you would avoid Alzheimer's.
There's always
So
this issue of, is it correlation or is it causation? It's like with
yeah,
cholesterol, for instance. For a long time people said, well, high cholesterol
yeah,
causes
yeah.
problems, but it's not clear that's the case at all.
That's
It
right.
depends on
Right.
whether
Well,
they're oxidized and whether you've
yeah,
got
yeah,
lesions and so on.
right, right. Well, I mean, I was reading about a drug today that a company has testing that improves uh, a lipoprotein 1 receptor in the brain, it boosts it up, and that causes a decrease in the amyloid to get clumped, and it gets rid of it, and it raises the amyloid in the bloodstream, but it reduces it in the brain by 40 to 50% in the animals overnight. Incredible. And, it's not yet quite available as a drug for human trials, but it'll be here soon. I mean, it looks pretty safe. And, and that's just one of a handful of companies that are really on the cusp of having something that'll be available.
Well, you're doing great at 80. You're looking good, and your brain's functioning well. So keep that up.
Thanks.
Check back in a couple of years and see how things are going, shall we?
We'll do that. Thanks a lot, Max. Enjoyed it.
It's good talking to you.